Good morning, good afternoon, and good evening to all participants. Thank you for joining today's webinar, Adventures in DMPK, from small molecules and PROTACs to peptides and antibodies. With that context, I'm pleased to introduce today's speaker, Dr. Justin Cui, who will share his experience and technical insights on DMPK strategies across small molecules, PROTACs, peptides, and antibodies. We encourage you to submit your questions in the Q&A box. Toward the end of today's section, we will have a couple of minutes for discussion. Without no further action, Dr. Cui, it's over to you.
So I'll share the first overview of biotech structure and basic drug discovery. We have over 10 years, more than now, I think it's 17 years since the company established. And we have a variety of milestones. For example, is listed IPO in 2019 and acquired another company. We have a CDC manufacturing capability for small molecule. And back to the DMPK pharmacology platform. In 2009, we have animal facility licensing and have the animal facility established and doing the oncology CRO program and further supporting the international and biotech at home. And we also have the small molecule compound DMPK platform established. And then we have the CRO and supporting the VBI, our own company supported startups invested. And we are supporting this program very well. And it's developed into up to 100 Viva invested company. In 2002, the animal facility expanded in a new, brand new building. And last year, we optimized our capability in small molecule, antibody PK, tumor and autoimmune disease, and also metabolic and other biomarker study and pre-tox studies. So without further ado, I would like to dive into our topics outlined here. And we have the capability in small molecules, for example, small molecule in vitro and in vivo study, PK modalities for small molecule, PROTAC, peptides, antibody ADC, and biologics. And also, we have the immunoassay system. For example, the cytokine assays, TB activation, differentiation, and functional assays. And for efficacy study, I'll introduce the tumor efficacy evaluation, autoimmune disease inflammation, and some of the metabolic disease and diet-related drug. And I'll further introduce our capability in CRS, TA, and other aspects of our capability, and if we have time. First, I would introduce ADME capability and the PK studies. And for small molecule PROTAC and peptide, they have some similarities for the PK and ADME. So I just briefly introduce the capability we have right now, or the advancement in this field. So for R&D study, functional assay, PK efficacy evaluation, and tox and IND filing, this is a very important steps to develop new drugs. And for ADME in vitro for small molecule, and we need to first have the compounds generated, and for physical chemical property analysis in this field. and metabolic stability, and including the in vitro study and also in vivo study for the enzyme, whole blood, hepatocyte stability, and the fractional stability in liver sites, S9. And also the phase 2 studies for the small molecule metabolism, and also for next step for distribution study, and also later for the metabolic profiling for meta-ID study, and also for the permeability study and transporter studies in MDCK cells or CACO2 cells or permeability. The further PK study in small molecule is for measuring rodents and dog, and for PK study in biologics, especially antibody and ADC, is rodent and NHP. And for the PK study, discrete and exciting dosing, we can commonly use to have great experience on that. And for antibody PK study, is a little bit different from the small molecule PK. Is introduced into in vivo in mouse and rat, and eventually is also need a monkey study for the all relevant species in monkey PK and tox. And ADA is a neutralizing antibody detection, and sometimes it's also necessary for the IND filing. And we can also perform the cytokine release syndrome facts and RO receptor occupancy study. The last part is efficacy studies in tumor autoimmune disease inflammation. And we have the syngeneic or CDX model and in vivo imaging system, autoimmune disease, and also metabolic disease. I just briefly go over the PROTAC study. We have the capability and support money programs. And there are some issues in PROTAC analysis, but basically small molecule. And the evaluation of PROTAC is basically similar to the other, the previous that I introduced. And for the peptide PK analysis, there are 5 species or more species we can do. And we can choose a variety of administration route, sub-Q, IV, and intramuscle studies. And this is just an example. PK the 4 species. This is a sub-Q study, and you can check from this curve, and it's IV study in dog. And our experience from many years of experience for the PK study, for either for small molecule or peptides, the analysis primarily the same using vanillin, but from the past experience of drug evaluation, we just collected our data and analysis it for example, AUC, and also through the IV OPO, maybe the low bottom 10% if with a lower AUC, so we can cut off this and without further analysis. So in this case, we can give clients guidance for example, AOC also is applied in the T-half study and bioavailability analysis. And this is maybe over 100% of top 5. Maybe clients, maybe specialized want to this kind of bioavailability over 100%. Maybe we have experiences, maybe 10% of the total drugs have the bioavailability, so more than 100%. So in this study, we can give conclusion, troubleshooting, or guidance. Next, I move to the antibody ADC or biologics. So now is 4 classes of IgG. These have a different structure, especially in Fc part. Here, I just copied this from the immunobiology. So this is typical IgG1 with hinge region and Fc, and IgG2 and IgG4. Usually for the immuno-oncology, we use IgG1, which have the effector functions, and with IgG2 and IgG4, they have a hinge region with a disulfide bond, and this one is with 2 disulfide bond, and with the Fc function is reduced effector function, and using change the class switch into the engineering to IgG2 or IgG4. The half-life in human is maybe 3 weeks, or a little bit more, but this one with a long neck, and it have allotype dependent functions. So usually we don't use this form. This have the full dependent function of Fc effector function, and this one will reduce the function. So example for the LALA mutation, and also is from IgG1 to IgG2 or IgG4. And further, we can use the restriction Fc binding by the engineering or post-protein modification, or using engineering FcRn to evaluate in vitro for PK or in vitro PK study. So here is the FcRn effect, and also the IgG pharmacokinetics. And with the YT mutation, and is enhanced using these 3 amino acid mutated into YTG, tyrosine, and some other hydrophilic amino acids. And this will enhance its Fc binding to the FcRn, and to maintain the longer, make its T half longer. This is a marketing antibodies, some of the name or abbreviation. We fully use this advantage or mechanism and to check in vitro for the PK. There are publications, but we modify this technology is with a great advantage. Just basically give an explanation about this one. And A is the inner chamber, and this outer chamber. We use the MDCK for small molecule analysis, and put a layer of the MDCK cells, make is transcytosis from solution B to solution A. And for the antibody, we just engineered this FcRn B to M, and to make it permeable to antibody. This is example. As you know, with pH 6 or 6.5, the affinity FcRn binding to antibody is very high, maybe a log or 2 logs enhancement. And with a pH 7 is a dissociation with very quick and with low binding affinity. So we generate the cell line, and after put in a layer, make a semi-permeable to small molecule, bundled to the antibody. After adding the antibody and culturing 2 to 24 hours, and the antibody will transcytosate into solution B. We take part of the solution for the antibody analysis. This is a generation of the FcRn humanized MDCK cell lines. These are steps. I won't go detail about this this slide cartoon. This is using after the using alpha beta chain into the cells And we checked the expression using the FACS analysis. So we found this pool is great. So we isolate 4 clones and check one of them get very great correlations for the characterization of the PK. This is FcRn engineered cell line, and this is a wild type 1. And with wild type 1, there is no antibody permeability to the solution B. And this one, with the time-dependent increase of the concentration of the trastuzumab antibody in the study we use is commercialized antibody. And we also check the concentrations in the solution A, and it's with a different concentration of the antibody. And we check the concentration transcytotoxicity into solution B, and is dose-dependent manner. And further, we check the using the transcytosis assays using the oxidation agent, and is blocking the or inhibit the binding of the antibody to the FcRn. And we found this is indeed decreased permeate into the solution B. So further, we analyze the variety of commercialized antibody, and we found this is FcRn trastuzumab, and this is a YTE enhanced, and this one is with the LALA mutation, is reduced the FcRn binding, and even with that YTE. This is a YTE LALA mutation and also fusion protein, and this one gives a very high permeability to the solution B. And we also check the MDCK with albumin, and this is how also how the binding. So this is how a great advantage application. And so it can be used in PK in vitro and for screening, especially for YTE. Recently, we can use this for the screening for the antibody for the inhibition of the permeability or reduce the capability. Sometimes we use antibody with a shorter T-half. So in vivo study can be for 1 month study, but in this study we can use many candidates at the same time, up to 20 or even more. And within 3 days, if we have a culture cell ready, we can get the results. Further, we tested the correlation between PK in clinical data and our cell-based data. So we found this is antibody listed, and this is a YTE mutation. It's a kind of antiviral antibody, and it is from published data is the PK is 71 days, and we got the correlation is very great. We got correlation is 8.4. Sorry, 0.84. So this is application for antibody and fusion protein as well, and we're using this technology for multiple companies or doing this kind of screening. Later, we also have the capability for AIDD, and we have the company, we have a department producing antibody and design and check the antibody characteristics. So this is a 3 antibody using structure modeling and after conjugation and generated the construct of ADC. And after the calculation, the 5, 3, 4, and this one aggregation score is very low. This is a medium, this is very high, and they predict that this is the highest antibody. So we checked the stability in plasma at 37 degrees for a couple of weeks culturing. And this is the three all the controls. This is the antibody sandwich with the antibody against the ADC or FC, and also FC detection. So we found this one after 2 or 3 weeks. Culturing the antibody remaining integrity is 75%. And this one is medium. This is the worst one. So it's completely correlation with the prediction. So I summarize the key factors affecting antibody PK performance. For generation of the antibody property is should be tested for purity, solubility, serum stability, and thermal stability with a TM and a PI value as well. And for its FcRn is contribute a great deal for the PK performance, and also TMDD, target-mediated drug distribution, and also effector functions. Also, the immunogenicity, ADA or neutralizing antibody, and the enzymatic metabolism also contribute a lot. And cellular uptake, and also some typical clearance on some kind of disease, and loss of the inflammation in GI tract, loss of the blood, etc. So I'll focus on next step about the immunoassays. The cytokine assay and T-B cell activation and differentiation, and also functional assays. Nowadays, the immunoassay is a conventional studies, and we have about 7 years of experience on lymphocyte activation and differentiation, and also T-cell activation, T-cell signaling functional study, for example, T-cell mediated toxicity and bispecific antibody or by TCE in T-cell editing, and for example, in primary cell T-cell or activated T-cell, and B-cell polarization as well. And for innate immune cells differentiation and functional assays, and this include DC, NK cell, and neutrophils, especially antibody-related toxicity study, ADCC, ADCP, and also CDC studies. And now antibody is major in most of our efficacy evaluation, especially in autoimmune disease and to oncology. And even eventually antibody probably need, if monkey is a relevant species, it will be tested in monkey for PK and also tox as well. For the signaling, its needs are for signaling transduction in STAT phosphorylation and kinase array, where we can perform this as well. And the transcription factor assays also a reported assay as well. And cytokine study for intracellular, extracellular, MSD assays, and the CBA assay, and multiplex studies. This can be performed in mouse, rat, and monkey as well. So part 1 is immune cell activation related to the T cell, B cell, and NK cells. I'll skip this one, and probably is mentioned earlier, is not much difference. The T cell especially, we can variety of activations, and also the B cells. Signaling is just an example here, and probably the close to the T cell activation, some of the type of activation or maturation. For case study in functional assay and drug evaluation, and first we're going to the binding or blocking depends on the functional assays or requirements for the antibody, and the internalization as well for ADC studies. We can do the FACS DT3C cell viability assay and internalization and pH auto analysis and using the in-Q site. Recently, we investigated a variety of antibodies using either microscopically or using the in-Q site machine. Cytokine analysis, example for ADC and cell viability check, and using the bispecific antibody and targeted labeled with luciferase. Here is introduce another functional important for the antibody evaluation, ADCC, ADCP, and CDC. We check that using the PBMC or NK cells. And also using the JERCAT FC gamma receptor reporter cell lines, and also ADCP using macrophage, monocyte-derived macrophage, and also CDC using the human normal serum, or also using the rapid blood cells for analysis the CDC. For the next part, focus on the efficacy study in IO cancer, autoimmune disease, maybe metabolic disease. Now we have greater capability in tumor efficacy evaluation and using the syngeneic model and also CDX model or IVAS for metastatic or also topic evaluation for the tumors. Here is just give example of using this antibody. We're using N97 cell lines, a breast cancer cell line, and this is the antibody evaluation. And with for three doses, we can give a greater TGI inhibition. And using this as a syngeneic model and using different varieties of small molecules, we can check it's a great inhibition for the TGI. And also, it can be tested for the mechanism study until FACS analysis and collecting the tumor cells and doing the FACS analysis and either B cell, T cell, or other innate immune cells, and further analysis of cytokines or immunohistological staining for the proliferation growth marker, and also some tumor antigens, OTA. So I'll switch gear to the autoimmune and the inflammation disease models. Here is a list. I think a majority of this one, on this page, we have finished the evaluation and some of the models for client services and project. I'll summarize our capability. We have one-stop capability for the later stage of drug analysis for small molecule peptides and PROTAC, antibody PK profiling, ADA FACS analysis, and antibody distribution studies, and the PD cytokine profiling, and in variety of species, and doing the antibody PK and the evaluation. So this is my last slide for the presentation, but I'll show a couple of companies' technology platform. So we have AIDD and CIDD platform, and can help us de novo design, and also doing the for peptide, for antibody affinity maturation, and also antibody production department. We collaborate closely each other, and moving many integrated projects moving forward. And we will also have the global localization. In China, we have several parts, and also we have a site in Boston and the UK and other countries. So we have many clients around the world, especially in US. Thank you very much. This is my last slide, and